Rajesh, you can also start taking the attendance as you are taking before.
Yes, sir.
Everyone else can go into the mute mode. I will also switch off the video once I start giving the lecture. We'll wait for 1 or 2 minutes. Participants, I can see how many have joined. Yeah, 43 have joined only. Okay, can you see the slide, everybody? Rajesh?
No, sir, not yet.
Okay, let me see. Yeah, can you?
No, sir, not.
No?
No, sir.
No, sir.
Okay. Sharing. Stop sharing. Is it okay now?
Yes, sir. Visible now.
Yes, yes.
Okay. Uh, Welcome everyone to this part of this course UC 206. I am Tushar Kanti Chakravarti, TKC, and I will be taking this part. This is on chirality part of that course and symmetry elements. And I've tried to break up the course the way I have given. So today it will be only I'll give introduction that why we need to learn chirality, and some of these things like a RS configuration and symmetry elements you might have already read in your intermediate, but it is better to go through them once again. And then we'll talk about various kinds of stereoisomers, and then we'll talk about optical activity and one of the most important aspects of stereochemistry. And then we'll talk about chiral resolution. We'll talk about stereo selective and stereo specific reactions, and then at the end we'll talk about conformation. And as you can see here that the final exam of this course will be only on this part of the course, because whatever you have learned in Professor Biju's course that has already been taken care of, and this will have 50% of the total marks of the like a course, and we'll have about 3 assignments for you and maybe 2 quizzes, so which will roughly about 60%, and final exam will be 40%. So total will be like 100%. Out of that, 50% marks will be added to Professor Biju's part, and then gradings and all those things will be done. So tutorials, if you have done before, so it will be compulsory for all of you to attend the tutorials, and whatever I teach, all the class notes, all the slides will be available to you from the next class onward, and you can go through only those notes, and that will be sufficient for you to learn and also prepare for the exam. And but those who are interested, I don't know whether you are in campus or you are at home, whether you can have access to these books or not. So 2 books are good. We have used it possibly in our student days, and they are still available. And it's like a Dhananjay Nasipudi's book on stereochemistry of organic compound. This is a good book. And other one older book and still available. I think the new editions are good. Stereochemistry of organic compounds by Eliel. So these are the 2 books, but you can possibly consult any book or Google or any other thing. So what I teach is not exactly taken from any of those 2 books, but you can find all those things in the those 2 books I have written here. So I have downloaded many of those things from the internet also. So you can also use internet or Google to find out what I am teaching if you want to learn more about the subject. And as I said, like in this course, you don't have to memorize anything. So I will not pressurize you to memorize different aspects of the chemistry. Whatever is taught, like a basic concept should be clear to you, so that any question I ask, you should be able to answer. I can see one hand is raised. I don't know why. Any problem?
Yes, sir.
Yeah.
Hello. Yes,
Yeah.
sir. So in the slide, the
Yeah.
given final exam will have 50% marks, and right below that you said it has 40% marks. It's
Total like a 60 plus 40 will make it 50%. I mean, like a 100% out of 100, I will make it half to add it to the previous component. So this 50%, 40% of that 50% will be from the final exam, and 60% of the 50% basically will be from the assignments and quizzes. All right.
okay. Okay, sir. Got it.
Okay. So anyway, so as I go along, so I may not be able to take all your questions, what you have doubts. So you wait for the class to end. Maybe I'll stop 10 minutes before, so that those of you who have some question, you can ask those questions, or you can clarify your doubts during the TA classes. You can also email me, and my email ID is written here, tushar@iiscse.in, or this is the two ways you can communicate with me. And basically, like just relax, and then let's see what we learned from this course. We will have about 12 to 14 lectures in this course, and I'll let you know beforehand when we will have the quizzes, so that you can prepare for those quizzes. And this class, as I said, will be like a general introduction, not starting the subject matter. So again, Pranav, you have raised your hand. If you have any question, you can ask, but you can also wait for the class to end.
This is just about the logistics. So it would be helpful if you could, if you're using pre-prepared slides while if presenting, it would be helpful if you could upload them just a day before the class or so.
Yeah, no, the whole courses, all slides will be uploaded before the next class. And so when I go through the slides, when I present, explain, then you will see the slides. I will tell, slide number 1 or 2, something like that. So you can check your class notes also. So that will be done. Whole course will be done in 2 batches. First, I will this week I will upload 1 lecture, and the second one will be as we go along, a second note. So everything what I teach will be from those notes. It's about 110 or 12, 15 slides will be there total. All right.
Yep. Thank you.
So, but not from today's lecture. Today's lecture is like a general introduction, so you don't need to go through it for your exam. So you'll get confused. So I am not uploading this one, and so don't worry. So we'll be all there to help you out, and I don't foresee much of a problem as we go along. So why we learn stereochemistry? So this is very important for us to see. And let us see if I can sling here. Yeah, molecular chirality. You can see that basic building blocks of all living organisms are chiral. Now we human beings are composed of biomolecules. You know that we have DNAs, we have RNAs, we have the proteins, and many other molecules. Basically, we are human beings are all made of molecules, and all the molecules are mostly chiral. For example, all the proteins are made what we call left-handed amino acid. So these are natural amino acid. And what we call right-handed sugar, like D sugars, glucose, pentose, and many other things. And so all these molecules are chiral. And chiral molecules in living organisms in nature exist almost exclusively as a single enantiomer. So you don't find any molecule in the body. Suppose you have a protein which is chiral, you don't see its other enantiomer in the body. It has only one enantiomer, and in the a very pure form. And this recognition of the chirality, molecular recognition, and many other things is basically the prerequisite for the origin of life. And as you can see that when we synthesize molecules in the lab, in absence of any directing template, that means you just mix 2 reagents and some reaction takes place, and if you generate one chiral center, normally we get, most of all the time, if you have no chiral template, then we get a racemic mixture. So that means both the enantiomers you get in equal amounts. But in biological molecules, all these things are made as a single enantiomer. So like even some of the amino acids, for example, are prepared in our body. All the amino acids are not required, prepared. Some of them are, made in our body, and they are also made in the chirally pure form. So you can ask that what started, when the life started, how did the chirality come? Because there is no chiral environment, billions of years ago in the Earth or any other planet system. So how does the chirality originate when there is no template? And how this, once it started, of course, there are many theories are there. Large number of people have worked to find out what really started, the chiral environment. And then once it starts, how that bias gets sustained and propagated to give all the biological systems and the living system that you see, which are made of chiral molecules. And so there are many theories. There are some people say that there is physical asymmetry that stays in the interstellar space. For example, light polarization may have degraded the d enantiomer more. So one of the enantiomer might have got degraded more, leaving an excess of one enantiomer, and that those molecules got transferred to the Earth by comets, asteroids, and meteorites, and so on. Because some people have collected molecules, for example, from meteorites, and then found that they contain sometimes some chiral amino acid. So this could be one of the theories, but there is no foolproof for any of those theories. And some of the suggest that favorable contribution from D sugar leading to the chiral discrimination. But then the question is, how did sugar get the chirality? Okay, whether sugar came first or amino acids came first, that is also a conjecture. Sometimes we see that when you make a dipeptide, for example, racemic mixture, and there is a tendency for a molecule. Suppose I have an amino acid which is a mixture of equal amounts of R and S enantiomer. If it has to combine with another amino acid, then it prefers to combine with the opposite stereochemistry. So if R combines with another amino acid, it will combine with the S. And then R and S, you get a dipeptide, and S has RS has a tendency to pick up R. So you are already introducing some bias when some of the peptides backbones are getting formed. So there are like many theories, but whatever it is, the result is amazing. As I have written at the bottom that homochiral polymer of n monomers, and you have one isomer if you have to go to 2 to the power minus n, and a small protein with, suppose you have 100 amino acid, and that probability becomes 2 to the power minus 100. So but you get only one isomer, and so the end result is very amazing, and the way evolution has taken place and today where we have reached. And let us see. No definition of chirality. So like I have written some of the structures here, and L-alanine, and then you can have a mirror image which is like a D-alanine, and if you can write also the structure in different formats. So I will, as we go along, we'll try to learn how to draw a three-dimensional structure on a piece of paper so that you can get the idea about the three-dimensionality of the molecule. And so all these, and if their mirror images are same, superimposable, then they are not chiral. For example, only one of the amino acids, like a glycine, you can see the attached to 2 hydrogen and then 1 amine and then 1 carboxyl group. So this molecule is not chiral. So all other 19 amino acids, natural amino acids, have a chirality as shown with example of phenylalanine. And is a direct consequence of the inherent molecular dissymmetry. So if there is a symmetry, then this is not a chiral. So if there is a inherent dissymmetry in the molecular structure, that gives rise to what we call chirality. And how you know if a molecule is chiral or not? Of course, if you have a three-dimensional model of the molecule, and you make them that molecule, and you can make its mirror image also, and then try to see if they are superimposable or not. If these two molecules are not superimposable, then you can say, okay, this molecule is chiral. Or you have to look for some symmetry elements in the molecule, whether those symmetry elements are present or absent. And if those symmetry elements are present, then you can say, okay, this molecule is symmetric and this has no chirality. And if there is no symmetry element, then you can say, okay, this has an inherent dissymmetry, so this molecule will be going to be chiral. And you know that historically in Louis Pasteur, he first investigated the shapes of ammonium sodium tartrate. So it is a salt of tartaric acid. Tartaric acid structure is shown here, and then he could crystallize, you know, the two isomers, two enantiomers, and could separate them by hand picking, you know, the different crystal sizes. He was very, you know, fortunate because the condition that he used allowed these two enantiomers to crystallize separately. And normally it doesn't happen, because if you have a racemic mixture, you will possibly in more detail you can learn in phase rule, but I will not go into the detail. This racemic mixture sometimes they crystallize as a racemate. It does not crystallize separately. But under certain conditions, possibly when you try to, you know, crystallize, and that condition was favorable for him, and they separate out as a pure crystal in a pure enantiomeric form. And then he measured, you know, the optical rotation for these compounds and found out, you know, they are two different molecules. And that was in 1848. I think that was first reported. And to tell you, you know, like even the carbon has the 4 valency that it attached to 4 different substituents was not known at that time, because that carbon has a 4 valency was the first published by Kekulé. Kekulé, you have heard, who gave the structure of very famous molecule called benzene. So it was possibly in 1856 that Kekulé first postulated that, you know, carbon has 4 valency. So much before that, the concept of chirality was introduced by Louis Pasteur. And then, you know, after almost like 25 years, and then Van't Hoff and the Liebel, they, you know, gave that, you know, that stereoisomers of organic compounds, if they have n asymmetric carbon atoms, suppose for example you have a tartaric acid, it has 2 asymmetric carbon atoms, so that possible isomer will be 2 to the power 2, that means 4. So if you have n chiral centers of the molecule, then you can have 2 to the power n different isomers possible. So they slowly, you know, from there people started learning about the concept of chirality. And basically it means the handedness, you know, in Greek term it is called Kheir, K-H-E-I-R. So in Greek alphabet you can see that, you know, it is written like this. So it means that, you know, left-handed and right-handed. And as I said, like it is a challenging task for synthetic chemists to make molecules in the chirally pure form. And of course, you know, as I said, like nature makes all the time, and all these chiral molecules in the one enantiomeric form. So that is very amazing. And how does it happen? And some properties also of the molecules will be different. For example, this limonene, it's a natural product, and it comes from R plus limonene, and you can have S minus limonene. And this R plus limonene, if you can, you know, have a good, you know, way of smelling things, so you like it, it smells like orange in R limonene, whereas S limonene is smells like a lemon, more, you know, soury smell. And many other molecules, you know, then oranges and many other molecules have a chiral molecule which will have a different properties. And this is important, as I was telling, that nature makes these molecules in a one pure enantiomer, because nature uses many of those enzymes, and enzymes, as you know, are composed of proteins which are composed of amino acids, so they are chirally pure. And this enzyme, for example, here it binds to a substrate, and it carries out some, you know, chemical transformations, and this substrate then binds to this the binding pocket we call of the enzyme, And this is called, a term like a lock and key hypothesis, which was postulated by Emil Fischer. This term was coined by him first time. And this fits in very well into this enzyme binding pocket. And then enzyme carries out the chemical transformation. Suppose it breaks this molecule into two portions. And then, this substrate will then come out after this bond is cleaved or the chemical transformation has taken place. And then this enzyme goes around and then, carries out this reaction again and again, hundreds and millions of times. For example, if you have a peptide composed of one D amino acid, and if you have normally all L amino acid, suppose you have some D amino acids, and then that particular peptide will not be recognized by any enzyme. So that's why we said that to increase the metabolic stability of peptide, you can introduce sometime D amino acid, because the peptides get degraded very quickly inside our body, because we have a lot of enzymes which will degrade those peptides. But suppose I introduce some unnatural component into that peptide backbone, then it will not be recognized by the enzyme that we have in our body, and it will not get degraded so easily. And to give you a visual representation and what it means, that you have a molecule called epinephrine. This is a natural, a hormone kind of molecule we have in our body, and it has to go and bind to a receptor. Then receptor, you can see that it has a positively charged pocket, delta plus, delta minus, and this epinephrine will go and bind there. And this hydroxyl group is slightly electronegative, so it goes to the positive side. Then these hydroxyl groups here, like a phenolic, they're also slightly acidic, basic, and then they go to this side, and then it fits in very well. This ammonium component goes to the delta minus side, and it fits in very well. But if you make a chiral, enantiomer of this molecule, you go to the S epinephrine, then you can see that it does not fit into this binding pocket, because now you can see that you have reversed this stereochemistry here. Hydrogen is going down, and then then it cannot go and bind to this binding pocket, and the misfit with the binding pocket. So that's why this enzyme will not act onto those molecules. So chirality is very important, because as I said, like a lock and key theory, and it will be possible only for that particular enantiomer to bind to those binding pocket. And another example shown here, for example, this molecule, you have a chirality. So it is the cartoon representation. So you can see that this square portion binds to the square, triangle binds to the triangle, this one. But if you invert this center, so this rectangle comes here, then it will not go and bind to this receptor. For example, ibuprofen. So it is the structure of analgesic that all of you possibly take. If you have fever, you take some of the medicine, and I don't know, you take some medicine called Combiflem. Combiflem is like a acetaminophen and ibuprofen. And this ibuprofen is like only the S isomer is active, and R ibuprofen is not active. So all the molecules, many of those drugs you take are actually chiral molecule, and then only that particular chiral component will be active, and the other isomer does not show any activity. I have shown like a different metabolism also, like a D and L glucose. I said like we are composed of the right-handed sugars, and for example D plus glucose, I have shown the structure, its mirror image. All the stereocenter different will be L minus glucose. And like when we take sucrose or glucose, then it gets metabolized, and what we call it is the source of the our body energy source. It is stored in the form of an ATP. It gets, I'll not go into the details of this, but the first step for the L-glucose is like a metabolism is phosphorylation by an enzyme called hexokinase. And so it is in the glycolysis pathway, very important for the hexokinase to like a phosphorylate the glucose, which it cannot do if you give L-glucose, because it will not be able to recognize L-glucose, and that's sometimes L-glucose was used as a low-calorie sweetener, and not anymore. But people who have diabetes, sometimes you get substitute for the sugar. So you use fructose, you use L-glucose and many other forms, so that it remains in your body for quite some time. Although sometimes they have to also test, like as sweet as sugar, because even if your test buds, will recognize the molecular chirality, and the binding pocket will be has to be comparable to the sugar binding pockets. And as you can see, no. What has happened? Can you hear me?
Yes, sir, we can hear you.
Okay, some problem was there. I don't know what. Anyway, if you can see, like in pharma industry, for example, many of the drugs that we use, as I said, are chiral, but many of them are not chiral, and they don't like working with like a racemic molecule and molecules which have chirality, because there are a lot of what you call problem in making chiral molecules synthetically. So that's why most of the industry, they prefer to work with a non-chiral molecule which does not have many chirality. And but there are many small molecule drugs, for example, which don't have any chiral substance. For example, isoniazid anti-TB drug, it is a very small molecule. Pyrazinamide anti-TB drug, it does not have any chiral center. Then you have aspirin. This is a very old drug, more than 100 years old, and it works wonderfully, because if you have headache, you take this aspirin, and within 15 minutes you are, get rid of the headache. Then metformin, for example, this is an anti-diabetic drug. So these molecules don't have any chiral center. But natural product, most of the natural products will have a chiral center, and the advantage of this, this becomes more and more specific for a particular target. For example, in this slide, you can see that as you increase the complexity of molecule, branch length and many other, then it number of receptors it binds to becomes very specific. It becomes less. And if you have a very small molecule, sometimes it may have some side effects. Then it can bind to various receptors. This one shows like an anti-cancer molecule called paclitaxel, which binds to an enzyme called tubulin. Very specific binding. So it will not bind on any other target inside your body. So it will have a less side effect. And as we go along, you can see that many drug molecules are like derived from natural product, and it has we call natural product as a privileged structure. So the success rate for discovering new molecule becomes more as you extract molecule from natural sources like a plant sources or secondary metabolites of bacteria and so on. This like a slide shows the structures of many natural products which are very successful drug. So don't get scared looking at the structure, but as you can see, all these like molecules have a lot of chiral centers. Erythromycin was like a very successful antibiotic, and many other component variants of the erythromycin are available. We still take them today. Semi-synthetic means you start with erythromycin and make those new versions of these molecules which are more active. Then Taxol molecule I have shown like a anti-cancer molecule. Penicillin, all of you have heard of very famous molecule, and this is also anti-cancer molecule. And daptomycin, these are like a antibiotic being used. Artemether is anti-malarial compound. So all these molecules, as you can see, are composed of many chiral centers. Here, for example, you can see the cyclosporin. It is an immunosuppressant, rapamycin, and like when I used to be in school in 1967. And at that time, the first organ transplant took place, and that was in South Africa by a doctor called Christian Bernard. He is no more. But at that time, there was no TV, no phone or any other thing. So all we could get information from the newspaper. And then I can remember, the Bengali vernacular, newspapers were saying that now people can live forever because the most important organ that you can have in your body is the heart. So you have a faulty heart, then you can have a replacement, and then you can live forever. But of course, the patient died within 7 days, and because that time there's no drug available, immunosuppressant. Because as you know that when you take another organ from somebody else's body, then your body will reject that organ, and it does not. So you have to suppress your immune system till it gets like a part of your own body. So most of the organ transplantation nowadays are very successful because of the drug like cyclosporin, which are composed of amino acid or molecules like rapamycin, and so it has become very routine, like liver transplant or many other organ transplantation. Many other molecules are shown here. So this is the like a essence of chirality. So natural products, they belong in one particular isomeric form. And two very famous molecules, ivermectin and artemisinin, because of this molecule on the left hand side is used for filaria, for the treatment of filaria. So it's infectious disease, parasitic infection, and artemisinin is for the treatment of malaria. And these people, they got Campbell, Omura for the ivermectin, and UU2 for Chinese scientists got the Nobel Prize for the discovery of artemisinin. So again, as you can see, this is the success of the natural product-based molecules. And now many places people are finding good application for ivermectin for the treatment of COVID-19, corona. And exact mechanism is not known, but it is basically like works on the glutamate gated chloride channel, ion channel, and then it causes influx of more chloride ion into the cell, leading to the death of the cell. But how exactly works in COVID-19, it's not known yet, but they found its application. And many other, but there are also like a synthetic chiral drugs. As you can see, some of those molecules shown here. These have, idea you get from natural product like amoxicillin, that all the time you take this antibiotic. And this is also derived, from penicillin kind of idea concept, and then you make slight modification here and there to make it a better drug. And some other molecules shown here. These are synthetic chiral drug, not from the natural product. And today, if you have a chiral molecule, it is almost like a mandatory that you give it as a single enantiomer. You can't give it as a racemic form because of this particular case, in pregnant women was given this molecule thalidomide, and then it caused, like a formation of deformity in the babies that were born. And thousands of babies were born with deformed limbs, and that's why they are called teratogenic. And then at that time, this was banned to use this drug, and later on they found that this molecule on the right-hand side is one teratogen, and this is actually the drug molecule. Its isomer was causing this problem. So now it is almost like a mandatory, as I said, like if you have a chiral synthetic molecule to give as a single enantiomer. Also, like it makes sense. Suppose even if it is not harmful to your body, but you can see that one half of the molecule. Suppose you take a 500 milligram compound. So you give 250 milligram of the compound unnecessarily because it is not required for alleviating the particular disease. And so, I mean, it causes, also body to get rid of that, unwanted drug, because all the molecules that we take after doing its biological activity, they have to get degraded and get excreted from our body. So, unnecessarily you are loading the patient with a, like a half of the unrequired amount. Here, this slide also shows some of the other chiral molecule, like ethambutyl. It is an anti-TB molecule. And SS enantiomer is 200 to 500 times more potent than RR enantiomer. And similarly, many other molecules. S-timolol maleate, it is a molecule, it is used for the treatment of eye disease called glaucoma. And this was the molecule, first chiral synthetic molecule that was introduced in India in somewhere in the early 1990s. And this was the first project I did for the industry when I started my career. We developed the technology and gave to the industry, and they started. So it was like a 10-step synthesis, not easy to do, but it was done to carry out these reactions in the industrial scale. And then propranolol beta androgen blocking reagent, which is also like a S enantiomer 100 times more potent than the R enantiomer. So you can see that only one enantiomer sometimes more active than the other enantiomer, and that brings us to the concept of the importance of the chirality. And as I said, like you reduce the total dose given. Suppose racemic mixture you give 500, but 250 milligram is not required. So you can give only 250 milligram to the patient, which is not required. So drug loading will be much less. Now, I've almost like I told you today, and I'll not go more into the details of why it is important to know chirality. But to enjoy this course, you need to get a feeling for the three-dimensionality of a molecule. You have to get a feeling for how to draw a structure, and of course, once you draw the structure on a piece of paper, so it will look like a two-dimensional. It does not look very much like a 3D feeling will not come. If you see that, like in India, we are not very good, drawing things in the three-dimensional form. If you see these two pictures were almost from the same period, and in the Mughal picture, if you see, to describe the three-dimensionality, they used to draw things, from a length, like on the top you can see the emperor sitting, and in the front side you have other people. So this shows the three-dimensionality of the. You don't get a very good, idea of the three-dimensionality from this kind of picture. Backside, like you can see some river or some boat sailing and all. Whereas, during the Renaissance, like in Europe, they learned how to draw three-dimensionality. And this picture, very famous picture, you can see that you can get a feeling that it has a three-dimensionality in the two-dimensional painting. And I don't know, whether we use some software called ChemDraw and Chem3D to draw, organic molecule structures in our lab. All the students are using. I don't know whether those ChemDraw or Chem3D programs are available to the undergraduate students or not, whether you can download it from our IISc server or not.
Yes, sir, we can.
Okay,
we can.
Very nice. So you can draw any structure, and or if it is not available, then you can, free programs are there from SED labs or many other sources, and then you can draw a structure, and then the same structure you can transfer to. Once you transfer to Chem3D, then it will you can minimize little bit some program, maybe somewhere down the line I will show it to you, and then you can get the feeling for the three-dimensionality of the molecule. And so to enjoy this course, you have to do little bit, the exercise, so that you can get a feeling for how a molecule will look like, in real time. And so next slide, this one is for your practice. You can try. I think this lecture is being recorded, so you can see this one. This we call, stereo view. And many of you, when you are a kid, you might have seen something called viewfinder, or you have a small device and you insert one like a what do you call pictures inside the picture card, and then when you look through the viewfinder, you can see a three-dimensional picture. I don't know any of you have seen this or not. And this basically has the same picture at two different places, and then they coalesce. Like initially you get you will not be able to the three-dimensionality, but if you practice it, then you will be able to see. But those are like a exactly, the angle and everything has been fixed. So you don't have to, make a lot of effort to see the three-dimensionality. Automatically, it will show the three-dimensional picture. And I don't know if any of you have seen any 3D movie or not. And for example, those who are in Calcutta, you can go to Science Center, and there is a big theater, and then you can see beautiful, three-dimensional movie. And there you will have to use a glass. So you wear that glass, and then you can see the 3D movie. So I remember, like almost like 8 years back, I saw one movie in a theater wearing that glass. It was nice. It's called, I think it was called Life of Pi. I don't know how many of you have seen. So it gives a three-dimensional feeling. And as you can see that when we draw the structure of a peptide or protein, these are called PDB structures. And these two structures I have drawn. And one of the structures, the angle, suppose you have an axis along this, vertical axis. So this is rotated by about 3.5 to 5 degrees, depending on the software that you use. And then if you draw those three-dimensional structure, for example, in Chem3D, you can have a stereo view. And then you have to, like practice it. Now, if you look at these two structures, and slowly you cross-eye, and then once you cross-eye, like squint your eyes, those two structures will coalesce into single one in the middle, and then you can see the three-dimensional structure, and it will start floating in the middle of these two structures, and slowly you will not see the other two structures on two sides, and you will see only one structure in the middle, and that will be the superimposing of these two structures and giving a 3D perspective. And so this is important for you to practice, so you can look at this slide and try to see and see. Let me know how many of you can see the 3D image. So little practicing will give you how to see the three-dimensional structure from this 2D drawing. So I'll stop here today. And next class, then I'll start telling you about the symmetry elements and basically all these courses. I hope you enjoy this course and understand the importance of the chirality. If you have any question, you can ask, and otherwise you can also, as I said, like during the tutorial, you can clarify some of these doubts. All right, Chinmay or somebody else.
Good morning, Professor. So,
Yeah.
do we require the extensive use of 3D drawing softwares during this course?
No.
Is it like we don't?
No. Just to like feel for this molecule that we'll be drawing. Also, like you have to learn how to draw three-dimensional structure. As we go along, we will show you how three-dimensional structure is drawn on a piece of paper. And I mean, it will be different way of drawing than what you have learned during your intermediate courses. So this is just to enjoy the beauty of the chirality.
Okay, sir, got it. Thank you.
Yeah. All right, then I'll stop here today. So we'll take it up on Thursday at 9:30. Bye bye. Take care.
Yes, sir.
Yes.